very much for the invitation. It has actually been a very educating exercise for me, both at this room and also at the bar last night. I had a very long discussion with James, very illuminating. I have to say I'm disappointed. And the reason I'm disappointed is usually I stand up in conferences on human genetics and human sort of aspects of genomic medicine and so on, and what I have to do is basically convince the audience that biology is important, assaying gene expression, proteins, understanding endophenotypes is crucial, and I don't have to do any of these things, so I'm actually losing my pitch, which is that I cannot disagree anymore with the audience. But that's great at the end. I'll probably have to find something else to disagree with you, or some of you at least. So what I'm going to talk about is how we can actually, in practice, bring these phenotyping that we can do with cells into initially in research and gradually into the clinical practice. And I think I just want to point out one thing from James' talk yesterday, that these do not have to be separate. And we should actually try to merge them as much as possible, because that's the best way to learn and apply at the same time in the same way. And I think what is also important is that while all the methodologies of doing medicine, applying medicine, finding drugs and so on have been successful to some extent, and nobody can argue that they haven't given us anything, because there are pharmacies that sell thousands of drugs that in some cases do something, there are ways that we can approach this differently, and we should be careful about, on one hand, being too radical, but also actually being too conservative. So here's a paper that it's not like a real paper in the sense that people weren't actually meant to say that, but there's a paper that actually argues that parachutes are not necessarily useful when you fall from 10,000 feet or 3,000 feet. You have to do a double-blind randomized trial to test this. And basically, what is this supposed to say? That you don't have to do this to basically put 1,000 people with a parachute, 1,000 people without a parachute, to convince somebody of the obvious thing that physics will tell you, that having a parachute will necessarily help you. And we can actually learn that from empirical data. And this is actually one of the parts of the abstract. Again, it's not a real paper. It's not that people actually meant to say what they're saying, but they're stating the obvious, that there's a problem with thinking in identical ways about all the problems that we have to do might benefit if the most radical protagonists of evidence-based medicine organized and participated in a double-blind, randomized, placebo-controlled crossover trial of the parachute. Tell me what of you will actually participate on something like that? Probably nobody, and what we will learn that we don't know, probably nothing. Actually, probably learn some kind of dynamics of how fast bodies go down when they fall from thousands of feet. But at the end of the day, what we need to do, I think the two lessons from just thinking about this problem is that, first of all, we have to think out of the box. We have to think a little bit differently from what we have been thinking. And these traditional ways are gonna sometimes be helpful, but also a barrier in the way we have been thinking. And the other way we have to think about is that we need to actually learn from empirical information that will not necessarily satisfy formal criteria. And as long as this kind of empirical information are enough to get us there, it's actually better than not considering those information at all. So this is a very psychedelic picture. I think that we should give this to Andreas to actually ask people and find the genetic basis of understanding this. But basically, this is a picture that was published in Science, showing that there's a human being, curious, probably scientist, trying to learn the book of life, which is one of the reasons why we have been actually criticized so much as human geneticists, because we actually have been claiming in journals and magazines and newspapers that once you sequence the book of life and you basically find all the errors in it, which is all the different variants, we'll be able to understand disease, which we cannot. But actually, this is important to get you some baseline of where people stand at the start of their life at the zygote. Much of this information, together with the environment, of course, defines how the cell functions and how this brings together the body through development and the function of the adult. Now, in order to understand the genetics of a complex trait, we need to understand what the trait is, what it means. And one of the problems with all these studies has been that we actually define the phenotype in different ways, sometimes in an unproductive way, just because it's simple. So for example, if you want to go to, let's say, a case control study for diabetes, what people do is that they'd say these are people who are diabetics. These people are a random sample. We have to realize that controls, in the vast majority of cases, are not devoid of diabetics. They're usually just a random sample that contain the fraction of diabetics that you would expect in a random sample from the population, which is already a barrier because if the phenotype is relatively high frequency, that reduces your power. The second thing is we have no idea whether the controls will develop diabetes. But the biggest problem is, how do you define diabetes? How do you define that somebody's a diabetic? Well, in most cases, this is a health system-based approach. There's a threshold of a bunch of measurements that people make by doing some blood exams or some other types of more sophisticated exams, and they decide that this person is diabetic, has to go into treatment. These people should not go, and much of this is a financial decision, especially if you're an NHS. These are purely financial decisions of whether you should go into a drug or not. And so that threshold is actually not a scientifically defined threshold, is a health system defined threshold that is actually used afterwards to define how we use the phenotype. But there are many underlying continuous traits that actually define how this threshold is being set. And if we had the ability to sample the continuous distribution of a number of traits, we will necessarily get much higher power to understand the genetic basis of the distribution instead of just defining the genetic basis of being on the left or the right of a particular wall, which means that basically you're trying to find the difference between people here and people here, as well as people here and people here. So you're mixing too many heterogeneous things. Okay. Too many slides. Oh. Okay, I think it stops. Oh, good. Okay, but the other problem is that we're trying to understand how DNA variation that you inherit when you're born, actually when the fertilization happens at the zygote, affects a phenotype when somebody becomes 50 or 60 or 70 years old. Imagine the cell divisions, the molecular interactions, and all the cells that are in between those two processes, plus all the time that has passed. So we have two dimensions that we're not even touching. We're not touching development, we're not touching all these interactions in adult life, and we're not touching time. And time is very important because a cell that appears in exactly the same, let's say neurons, which are not replicating, they're not dividing. If I take a neuron when they first develop versus the time when somebody's 60 years old, they're not the same. It's the same cell, might be the same cell, but they're not, it might be the same cell with respect to the physical entity, but they're not the same cell with respect to the properties of gene expression of that particular cell. And so if we don't want to understand this, we essentially do not want to understand the pathophysiology and the molecular etiology behind the disease. So we need some way of actually accessing both those dimensions. The other problem that we have is environment. We kind of want to pretend that environment is very difficult to measure. So we're going to do the rough thing of who's smoking, what's BMI, who is exercising, and so on. Very basic things, which we know have generally radical effects, but we're forgetting exposures that we never remember. Like, for example, if I ask you what you had for lunch the last week on specific days, I bet in most cases you won't remember. There are also aspects of environment that are completely unrecordable if you try to do this, even with a questionnaire, with having the patient in front of you. Like, for example, the typical, I don't know how many of you have seen My Big Fat Greek Wedding, when they say that I don't eat meat, so you should have lamb. That is actually the reality in some of the Greek villages. People consider that lamb is not meat. Meat is beef. And that is actually a very big barrier in communication, because when you put this question in front of a person and you don't want to have an education segregated population, you're going to get different answers even if the phenotype or the behavior is exactly the same. So what we need to understand is how to actually sample this environment in an unbiased way. And what I'm arguing, and I think it's probably not difficult to convince you, is that this particular environment is written in our cells. If one has been exposed to something, whatever that is, smoking, infection, whatever, and none of your cells in the body have changed their epigenetic or gene expression or their genome because of that exposure, is as if that exposure never happened. It just doesn't matter because your body doesn't remember it. However, as is the case for the vast majority of the exposures, if that exposure has changed epigenetically or the genome somatically in a particular cell population, that means that this is the memory of the body for that particular exposure. What does that mean? That means that in ideal situation, if I could sample the whole body, single cell by cell, including metagenomes and so on, I could actually get to the information that I want. It's a very difficult thing. George Search gave us some ways of doing this, as well as Rudy yesterday, but it's kind of the thing we're trying to get at. We will not get there, but we'll have to find ways of sidestepping and getting the shortcuts for this. Now, this is actually not a single line. There's a bunch of lines here and there. I'm not sure why they're not being shown. But basically, what this is supposed to show is that as we move to cellular phenotypes, and this is sort of accumulated knowledge to organismal phenotypes. So let's say, for example, if I take cholesterol and if I take diabetes risk, the ability to dissect the genetics is becoming more difficult, even for the most highly associated variant. So it's much easier to find the top variant for cholesterol than finding the top variant for cardiovascular disease. And that is because of the distance, the biological distance. But if I don't have all the lines, I cannot possibly talk about it. The other problem is that we're talking about gene-gene interactions. A lot of people have been thinking about this, a lot of arguments about should we do this, how much heritability explains. Well, we don't have to actually ask this many questions and try to explore the statistical space. What we need to understand is that what will appear as a genetic interaction, as basically a significant interaction term in the linear model, at the level, let's say, of a phenotype like diabetes, will somewhere in the biological space of cells or tissues appear as a combination of two simple additive effects. So what Andrea says before is very important. It is the phenotype that defines what is the model of the effect of a particular variant. It could be that this variant here has a simple additive effect to a cellular phenotype, and this one here a simple main effect to a cellular phenotype, but those two together have to be considered as an interaction to explain something that happens at the organismal level. And I'm going to show you a couple of examples, a simple example of how this is done. The other thing we need to understand is that in order to get personalized medicine, we cannot just sequence the genome, right? That has been said too many times. I don't have to convince you again about this. We need to understand the individual as an entity and put it in the context of a reference population. So we need those reference information to understand how an individual compares to the average of biological information now, not just reference genomes or copy number variant databases. So what we need to do, I think, and again, that's not something, a lot of people, I thought I would be the first to say that, but I'm very happy to be the 10th or something that said that. We need to sample the biology of the human body. And by saying that, what I mean is that we need to sample the germline genome. That's easy, we already do that. We need to sample epigenetic marks in as many cellular environments as we can. We need to sample somatic mutations, not just for cancer, but for other things. We have actually no idea how many times a pancreas falls apart because the majority of those cells carry a mutation that the rest of the body does not. That information, we are completely ignorant. And maybe it's a lot of times, maybe it's none of the times. And we need to record the state of the body at the time that we make this observation. So we can measure BMI, height, metagenomes, infections, and so on. And one can imagine that one day you would go into the clinic and instead of getting 10 exams of blood that take like a few hours to be given back to the doctor and gives you sometimes little information, we'll be doing all this because sequencing is cheap, sequencing will be fast, and these things will be relatively trivial. And once we know where to do this in accessible tissues like blood, we will actually be accumulating a lot of information. And then, of course, we will need statisticians to be sitting next to the doctors interpreting this information because it will not be an easy thing to do. Okay, now how can we do this? Because it's not really obvious how we can get from this very nice theoretical framework to being able to address these questions, especially from the research perspective. Now, in research, we can get tissues, so we can do biopsies. And tissues are great because they capture the environment of that particular individual, that particular time point, plus the information that we think is genetically determined. Now, the problem is exactly, the advantage is exactly the problem as well. You lose power by having the environmental information, and the environment that you're going to sample is much more the environment that was recent and transient than the environment that is a consequence of lifestyle over the last 10 years. we can have cell lines, so lymphoblastoid cell lines, fibroblasts. Now, these are partially capturing all type of genetic effects that are maintained after the cell line generation. They have a lot of the genetic information, but they're actually missing, they're not on either of the two ends. So by doing this, you're actually accumulating a lot of information for some of the cell lines, but it's not easy to get all the cell lines to be taken out of the body and from biopsies, because not all cell lines can be maintained in a stable condition. And now we have the iPS cells, which we heard about. This is an excellent way to actually sample information and differentiate eventually to a lot of different cell types, with the only caveat that the theory says that we should be missing the environment. That's exactly what we're trying to do. We're reprogramming them to forget all their lifetime, everything they've been exposed to, and go back to the zygote, pretty much. And by doing that, we're actually raising the environment. Is that good or bad? Maybe, well, it's good because we are actually increasing the power of genetics. It's bad because we're losing all the environmental exposures that we would have liked to have. Unfortunately, we cannot just subtract and store separately all these reprogrammed features that we are dealing with in iPS cells. Now, we can do a lot of assays. There's no way that one can just do all the assays that are currently available. You can do gene expression, proteomics, epigenetic markers. There's a huge number of chromatin modifications. There's an ever-changing landscape of what these mean and how they correlate, what are the functions that they're associated with. There are also cytoplasmic phenotypes that could be relevant to a lot of different sort of signaling pathways and metabolites. You could have actually cell-specific phenotypes that have to do with cell division and all these things. There's a huge number of phenotypes. And actually, in order to learn which ones we should use, we should do as many of them as possible, learn what is the minimal information that gives us the minimal amount of data that gives us the maximal information, and go from there to apply this to larger populations. We can also do this in two different ways. We can have the steady state, so we sample a cell and we say this is a normally behaving cell, and that cell is going to be whatever is the exposure in the body. But we can challenge them with drugs or infections or whatever, which actually brings up certain aspects. And this is essentially what pharmacogenomics is doing. That's why pharmacogenomics is so successful, because it's challenging with a particular agent, and that particular challenge brings up a higher genetic effect by essentially erasing background and noise, which is what has been shown with cell lines as well. Now, there's one big problem in thinking about all these cellular phenotypes, and that is tissue specificity. Now, if I ask around the room, what do you think is the relevant tissue for diabetes? Most of you would say pancreas, beta cells, islets, liver. Other people would talk about other types of tissues that are slightly more peripheral and so on. But I think, although you all know this, I think it's going to be very hard for you to remember that hypothalamus is causal tissue for diabetes. And I don't know if I'm surprising anybody because the initial knee-jerk reaction could be a surprise, but it is for a very simple reason. Because hypothalamus, there are genetic variants that act in hypothalamus that define appetite control, which affects BMI, which later affects type 2 diabetes risk. And that is actually a real association. FTO variants are associated with both BMI and type 2 diabetes. And if we did not know that BMI is an intermediate phenotype, we would be talking about FTO being a diabetes gene. We don't do that because we have an earlier phenotype that we understand better. But how many times are we missing the better phenotype, and we call something the gene for cardiovascular disease when it's a gene for basically something else that we don't know? There's a lot of different missing points, and basically all phenotypes could be associated with certain variants. We just have to understand the model in which they're associated. But tissue specificity is very important, and we need to separate the causal tissue, where the genetic variant is causing the difference between individuals, versus what is the affected tissue once you have the disease. These are not necessarily the same things. And it does not mean that affected tissues are where the variants are acting in order to increase the risk for a particular disease. Okay, so gene expression is one of these phenotypes, and I'm going to give you very quickly some sort of basic results about how we can go about thinking about cellular phenotypes. So one of these, so we can do gene expression to understand regulatory variation, to annotate regulatory elements, because essentially we're doing a mutation screen from nature. I think Ernst will tell you a lot about the mutation screen that we're doing in Drosophila by throwing mutations in Drosophila and then sampling the phenotypes. Essentially, we have a big screen now out there, and we're just sampling it. We can support disease studies by providing endophenotypes, like gene expression. We can assess the distribution of molecular effects in the genome. We can understand what exactly are the molecular differences among us, not just what happens with risk when somebody's 60, but how do your cell is different from mine and yours and so on. What is the first thing that is different between two individuals that, let's say, differ by one single nucleotide in the cell, in the development? That's very hard to address, but that's kind of an asymptotic goal. And at the end, we can understand about natural selection. We should not forget that evolution tells us something about our history and disease. It's not completely uncorrelated. For example, what is currently variable in the genome is probably what was variable, a million years or 2 million years ago when evolution was sampling from, either through selection or drift. And whatever is being sampled now will actually define how we look in 1 million years from now if we are still around. Okay. So my lab has been working for many years on discovering what we call EQTL. So EQTL stands for expression, quantitative trait loss high. So we're finding variants that affect levels of gene expression, either near the gene, so that these are affecting cis-regulatory variants, or in trans, these are usually basically variants in transcription factors that affect the expression of their target. But they don't have to be transcription factors. So we did one experiment back when I was still at the Sanger, where we took 62 individuals. We RNA sequenced with Illumina technology with paired-end sequencing, and we wanted to understand the association between the expression inferred from RNA sequence and the genotypes that we have for these individuals, which were from the 1,000 genomes HapMap individuals. Okay? The bottom line is, and I don't want to go into the technical details, is that if you compare the data you get from the same individuals, the same genotypes, but you do this between RNA sequence and arrays, you actually get twice as many discoveries as you get in RNA seq as you get with arrays. And that is because your resolution increases by a lot when you do this RNA sequence experiment. Because you're looking at sequence, you can find associations with fine-scale sequencing effects that you cannot find otherwise. But you can also access allele-specific differences. So you can tell the difference now between the two haplotypes because you actually see the sequence. And that gives you the ability to see it. This is showing terrible. You can see differences in alternative splicing between certain haplotypes. And you cannot, wow, okay. And you can also see that this is basically showing, I'm not going to explain it because it's going to be impossible. But essentially, we can discover regulatory variants that are not accessible by association, but they are accessible because we can look at the allele-specific effects closely with the sequence and the RNA sequence. And basically, we can do that in a population genetics way where we can see long haplotypes that are recent that have a particular regulatory effect, or by looking at a number of regulatory haplotypes that are associated with differential expression of the alleles, but are not necessarily affecting many individuals. That is very useful because those types of variants are not accessible by arrays because we cannot find the association and we cannot see them, and it's likely that these are larger effects and therefore more likely to have high penetrance, though they're not explaining much of the disease variants. We can also see evidence for nonsense-mediated decay. So stop-gain variants are actually likely to create a decreased level of expression because through the nonsense-mediated decay pathway. Okay, I'm going to talk about context a little bit. I'm going to give you a few examples of, although we can think of all these things out of context, of any particular sort of covariate or environmental or whatever you want to call it parameter, it is very important to think about what is exactly the setting that we're looking at all these things. So let's think about haplotype. Okay. It's going to take forever. Okay, so basically, this is, can you change the slides for me? Because I think I'm losing minutes trying to do this. Okay, next slide, please. And again. Okay, so basically the idea here is that if you look at the non-synonymous variants that are associated, that are found in the genome, just in certain number of individuals, and you ask how many of them that are in heterozygosity are actually 50-50 contributing to the transcript pool? And the answer is only 3/4. So 75% are contributing close to 50-50. That means that 25% are not. So what you would infer from the genotype is actually incorrect, because their contribution could be imbalanced to the point that you may have one variant that contributes 90% of the transcript while the other one contributes only 10. Now what does that mean? That if this is a variant that in homozygosity could be disease causing, if you just genotype that individual, you may not infer the potential phenotype, because you're not accounting for the degree at which this particular allele is expressed. Next slide. Previous slide. And we actually see that it has an evolutionary consequence. So if you look in populations, this paper just came out last week, we can see that, for example, protein coding variants affecting non-synonymous are actually more likely to be are less likely to be in the highly expressed phenotypes, are rarer in the high expressing haplotypes than the synonymous variants. This is really throwing me. Next slide, please. It's thrown me off completely. Okay, so the model is basically that we can actually define the penetrance of a variant, not by simply its presence, by combining the particular genotype that we see for this variant with the regulatory landscape of the haplotype in which they're found. And by combining these two, the risk prediction is going to be actually much more accurate than just taking one or the other. Next slide. Okay, tissue. Next slide, please. So basically, we have RNA sequenced three sets of tissues from 56 newborns from umbilical cords. The analysis of the array data for this has been published back in 2009. And now we're RNA sequencing to see whether the effects here of tissue specificity. Next slide. And we see that there's extensive tissue specificity of these regulatory variants. So that means that if I have a variant affecting expression in fibroblasts, the chances that this affects expression also in lymphoblastoid cell lines or T cells is actually less than 50%. So that means that if I'm considering function, I should consider it in the right context, tissue in this particular case, and therefore it makes it very difficult when we're looking at tissues like beta cells for diabetes or something to actually assess function in a reliable way and infer the biology that we want to understand. Next slide. We can look at alternative splicing effects. I don't have time because I lost time with the next slide. And the tissue specificity, we also see that it actually goes into the epigenetic effects as well. So epigenetic allelic variants are equally tissue specific as the genetic allelic variants. Next slide. Let's talk about sex now. And I'm talking about sex as a context. So we can do a gene expression analysis by looking at sex-specific effects. Next. Next. And we can actually do the association by combining individuals or by doing separate by sex. Next slide. And we see that there's actually extensive tissue specificity of these regulatory variants. Next slide, please. We can look at the distribution of where these are found, and actually this is biased. Next. And in some cases, we have genes that have both female-specific regulatory variants and male-specific regulatory variants completely uncorrelated. So basically, there's one gene that is differentially regulated by two different factors, depending on where you are in the genome, by males or females. Next slide. What we're doing now is we're actually, that's fine. We're actually going into extensive studies of cellular phenotyping by integrating initial trios and eventually numbers of individuals of a large number of assays that can be done at the genomic and the cellular level, so that we can understand exactly how gene regulation is progressing. Can you go ahead? Next slide. And we see we are doing various assays in the genome. Next slide. And we can see there are correlation. Next. And we can see that there's extensive allelic specificity. Either we're looking at POL2 binding or primary transcription, which is by this global random sequencing assay, or by looking at mRNA sequencing. And now we're starting to understand how the signal, genetic effect, is transmitted from here all the way here. Next slide. Now, you have a disease mapping locus. What do you do with it? How can all this data that are kind of glossed over can help you understand the biology behind this variant? Next slide. We have devised a method where we can actually integrate from two reference sets, so they don't have to be from the same individuals, expression or any other cellular phenotype association with a disease association. And there's a scoring function with 1 being a very high overlap, meaning that it's likely the same causal variant, 0 meaning that it's essentially no overlap. So they're completely independent genetic effects. Next one. And we see that if we apply this in data from EQTLs from 3 different tissues, from the mother study, which is a twin study for gene expression, we see that we have enrichment of 1s or close to 1 scores when we look at the genome-wide association studies that are currently in databases. So we can identify a number of these variants and actually generate some testable hypotheses for the biology that is mediating these particular effects. Next slide. What we see actually that is very interesting is that the tissue specificity of these signals is slightly higher than we normally see for the average expression QTL out there. So that means that disease variants are acting in a more tissue-specific way than the average functional variant out there that is not causing disease. Next one. Next slide. And I'm going to give you 2 slides of what we think is becoming a successful story. So this is KLF14. It's a gene that has been associated with type 2 diabetes, HDL cholesterol. It's a part of origin effect, and we know that it goes to type 2 diabetes likely through insulin resistance, part of origin specific effect. Next. We have identified transregulation of about 40 transcripts in subcutaneous fat in the Mother Consortium. This in collaboration with Panos De Lucas, Tim Spector, and Mark McCarthy. Next slide. And we have shown by integrating the data with the data from DECODE that these effects are actually of maternal effect, of the same type of effect. Next slide. So what we can do actually is try to build a story. Next. So we can go from variation near the KLF14 factor that affects expression in fat to with trans-EQTLs. We can go under a number of other genes. Next slide. These genes, expression in fat is highly correlated with insulin-resistant phenotypes. Next slide. Next. And several of these trans-EQTLs are strongly associated with those insulin-resistant phenotypes. And what we're starting to build is basically a story by integrating gene expression, association, multiple different tissues, a story where we start from an EQTL, a variant that affects gene expression, to understanding how likely what it appears to be a signaling mechanism from fat to other tissues to develop insulin resistance. And there's a number of experiments that Mark McCarthy and we're collaborating with in mice and other types of models are being built around this particular model. And what is appearing is that by just simply integrating in what is currently a naive model, we can start building biology in something that otherwise seemed like a lot of pieces that were disconnected. Next. Next slide. So in summary, genetics and cellular phenotypes are important to understand the molecular etiology of complex traits. RNA sequence, as well as any nucleic acid-based assay, because of these prices and technology in sequencing, offers great opportunities because it gives us much more power and the ability to explore rarer effects that we wouldn't see otherwise. We have a lot of context-dependent effects, and these are just things that we know. There's a lot of other types of things that are context-dependent. You can think of sex as a genotype, gene-by-gene interaction, right? Because it's a genotype effect. And we can use expression QTLs and other cellular QTLs to provide testable hypotheses for the mediators of the GWAS signals. So instead of having these 2 ends of the zygote genotype to a phenotype at 60, you can start building that, the transmission of that genetic signal and then interaction with the environment in order to be able to understand how you get there and how you can use this both for prognosis as well as diagnosis and treatment. And the last slide with the acknowledgements. Many people have contributed to this work, and thank you very much, and apologies for all this slowness in the slides.
Okay, we've got 10 minutes plus maybe for discussion.
Thank you very much for this brilliant talk, really. I'm so happy that you stressed the importance of context now in biological terms. So because of this, what do you think is the real value of IPS? Because you really do take them out of context. And of course, you have some advantages. But what is your interpretation if you if you identify a very important gene variant associated with whatever trait, and you don't see any consequence of this variant in iPS cells, electrophysiology, molecular biology, whatever, what is your interpretation of this?
Well, I think that one needs to be both careful about rejecting that this variant does something simply on iPS assays, because there could be lots of reasons, including things that you cannot understand in the sort of the iPS generation. So it's more of a confirmatory than a rejection tool. I mean, context is important. The problem is that one has to sort of value where exactly we need to invest. And if somebody tells me that tomorrow or sometime they can develop good models of sampling tissues from individuals, getting cell lines that we can do assays, we can get this in quantities that we can do ChIP-seq experiments in multiple transcription factors and chromatin modification factors, then I would say that's a good alternative, but I have not seen the possibility of that. The iPS gives us the possibility of getting to the tissue context. It's another aspect of context that it's hard to get with the tissue. So I think that we don't, that's why I mentioned all these three categories. I don't think that any of them, tissue, cell lines, or iPS, is the panacea to get to the question. It's basically all of them together. We need to be able to access as much as possible. And as we heard yesterday, biopsies are becoming much more easily accessible, but still there's not a lot of material. The cell line component is the thing we're missing. If we can get cell lines from the most of the biopsies that we have, that would have actually a good intermediate to get to these kind of questions that you're talking about.
Ernst.
So you also stressed the importance of the context and the cellular environment. In this, have you done, or do you know of any other people who have done studies that actually now looked at the longitudinal projection of expression EQTLs, so sampling the same person over 2 years or so, and seeing how that actually changes and what you can reflect from that?
So it's hard to do that because expression studies have just gotten to the point where we can have enough individuals, let alone having the sort of the time dimension. We have seen one effect that is actually time dependent, and it's actually, it can tell you a little bit how biology is complex. Well, it is complex, we just don't appreciate it enough. Some of the sex-specific effects that I'm talking about are actually aged by sex by SNP. So basically you have a sex-specific effect that you can see and replicate in adults, but you cannot see and therefore replicate when you're looking at 7-year-old children. And so that means that you have a three-way interaction now, which is essentially a nightmare to dissect. Imagine if you're starting to have three-way SNP interactions. And these are actually large effects. So basically by accounting for this, you would either ignore it if you were looking only at children, or you would find if you're looking only at adults. And it makes a difference because the hormones have not yet sort of kicked in when in a 70-year-old child, but they probably are in a much sort of stronger effect in later age. So we see time-dependent effects. Now, dissecting them will take a lot of samples. And I don't know if we can get them.
So if we play this in the future, does that mean in the future, and maybe in 5 to 10 years, newborns are going to be like they're going to be genotyped for certain rare diseases? Their genome is going to be sequenced, and they're going to be taken blood samples, say for example, and so the entire expression array in the blood is sequenced at various intervals until the adult. Is this a scenario that you can envision?
I think this is a little bit unrealistic because once you start to make them a condition that everybody has to do this in a time series, basically, you know, every year people go into the clinic and do a gene expression of whatever the tissues you can access, I think becomes a logistical nightmare. What I see more is that we can do some basic information that we know is there at birth. That's genome, right? You can sequence the genome because you know that is there and is going to be with you for the rest of your life. And then all these genomic assays will come at a point where you have a query. Like you go to the doctor because you have something, and at that point, this will be inferential. So you basically, you sample genomic information, trying to infer back, because I think that's going to be easier than trying to store all the information long-term and kind of follow it up. It's a purely sort of pragmatic approach. It's not necessarily the approach that, I mean, if we could do this, that'd be great. But I don't see how it would be easy to do this in a longitudinal way, unless you talk about a research project. I mean, a research project, I definitely agree we should do that. But for everybody that is born in Switzerland or in the world to do that, it might be a hard exercise. I see you disagree.
Well, I simply disagree because it's already now that people go to the doctor and they measure their blood pressure and their glucose levels in the blood as a preventive measure. So why
True,
not do that with
birth.
more molecular phenotypes, endophenotypes?
But not at birth, right? What I'm trying to say is that I'm not saying that somebody goes in a critical condition in an emergency room and that's when you do these assays. But when you're actually getting into the space, the age period where you start to worry about yourself and you start to have to check whether something is wrong, then genomics will kick in with all these extensive assays. I don't see how you can implement this from at sort of year zero. But maybe, I don't know.
Well, I think... Sorry, Effie.
Right. Can we ask if there's any other question? We have 1 more minute. Maybe you can continue over coffee. Is there any other question? Last question before Ernst and Manolis go on? No? Okay. Maybe then you should go on for a couple of minutes. There's nothing else. Okay.
This is a statement I made that already yesterday, and this is, if we're talking about bringing down healthcare costs and recognizing diseases earlier, I think one of the things that is not from me is from Thomas Goetz, who's written a very influential book, The Decision Tree, and he's mentioned that the importance of feedback control, and he argues that the only medical professionals who have actually, really got it to change behavior are the dentists, because we all floss teeth, we all brush our teeth, and because we know something is going to happen. And I think this kind of sort of more rapid feedback control is something that we have to build into our system. And so the more we know about how our values, whatever molecular QTLs those are, develop over time, the better it is to say something. And I think this is tells us more ourselves and makes us more change our behavior, as we heard yesterday, than a label on a package of cigarettes.
So I don't disagree with you at all about this. What I'm trying to say is that, I mean, if you go to the dentist, if a child goes to the dentist where they're like 15, right? I don't think they'll have an X-ray to every tooth, right? And that's exactly my point. That once you're in a position where it's an overkill to do all these things at a time where, in most cases, it will tell you nothing, then it's a little bit problematic. Like, for example, so my father is a doctor, and one of his, when he's whining about the young doctors, in justifiable or not ways, is that he says that when I was a clinician, simply because they just didn't have the tools, they would just basically get the disease from just getting the symptoms and not doing 2,500 exams. They would do just 5. And they would just guide themselves through, primarily because they didn't have the tools, but also because they were building the experience. Now, the criticism, I think is partly justifiable, partly not, is that you go to the doctor and you do all the possible exams that you can do, just in case you can find something. So what I'm arguing is that we should not do the same way we don't do a blood exam to a 10-year-old that goes to the pediatrician for cholesterol levels because we don't expect there's anything wrong with it. The same way we should not be doing this overkill when we don't think there's going to be an effect. But when you have risks identified at the genome level, of course you should follow them so that you can see how they progress, because you have a prior. And there I fully agree with that, as well as I fully agree that we should get into genomics and a large exploration as soon as we have any evidence that will inform us about changing decisions.
Thank you.